Welcome to our review on enzymes. So first thing we actually need to know then is what enzymes actually are. And the key phrase to remember here is that they are what's called a biological catalyst. So what that phrase actually means is that they're going to speed up a chemical reaction, but they're not going to be used up themselves. So basically a very small amount of enzyme is capable of catalyzing or speeding up a large number of chemical reactions. Enzymes have something called an active site. Now this is the most important part of the enzyme as far as we're concerned. So the active site then has a very specific shape. And what happens is that that shape of the active site fits perfectly with the substrate or the chemical that it's going to have the reaction with. So what we then find is that when the substrate molecule fits into the active site, then it brings together those chemicals that are required, and then they can form a bond to make a bigger molecule. Even though lower down the school you've only ever learnt that enzymes break large molecules into smaller ones, they are also capable of building bigger molecules. So just bear in mind that enzymes will either break large molecules into smaller ones or build large molecules from smaller ones, so it can do the two things. Enzymes have what's called optimums. So what we actually find then is when we're looking at temperature and pH, each enzyme has a particular temperature and a particular pH that suits it perfectly. And at that point, it's going to be working at its fastest rate. And that's what's called its optimum.